So today we are going to start the next topic, that is cephalin, or also known as phosphatidylethanolamine. I don't know we have studied cephalin earlier or not, but we are going to start from cephalin. Cephalin is also known as phosphatidylethanolamine. This is the basic structure of cephalin. The structure is similar to that of lecithin, that is on carbon number 1, R1 R2, whereas R1 जो है वो रिप्रेजेंट कर रहा है जहाँ पे फैटी एसिड ने अटैच होना है. R2 also represent where the fatty acid has to be attacked. This is blue, in blue this is the glycerol, this is the phosphate group, and with the phosphate group you can see that जो attach a group, that is ethanolamine. So cephalins are structurally identical with lecithins, except that the base choline is replaced by ethanolamine, as we discussed earlier. Cephalins also resembles lecithin in their physical properties. Cephalins contain more unsaturated fatty acid as compared to lecithin. So generally and by and large, cephalins are structurally identical and they are very much similar to lecithin. The main difference is that cephalins they contain more unsaturated fatty acid if we compare them with lecithins. Cephalins can be more conveniently prepared from brain, liver, and yeast. Cephalins are also found in most cell membranes, particularly in brain tissue, and they are also considered important in blood clotting process, as they are found in blood platelets. As we said that cephalins जो हैं इनको अगर हम prepare करना चाहें, extract करना चाहें तो conveniently we can extract them or prepare them from brain, liver, and yeast. Different studies have proved that cephalins जो हैं they are found particularly in nerve tissue, such as the white matter of brain, nerves, neural tissue, and in spinal cord. Whereas lecithin is the principal phospholipid in animals, whereas cephalin is the principal phospholipid in bacteria. So, what is particularly the role of cephalin in bacteria? It seems to be uncertain and not very much clear. Essential for life, ethanolamine, that this is the structure of ethanolamine, and ethanolamine occur in every cell in the human body as the head group of phosphatidylethanolamine, and it is present as free ethanolamine at varying concentrations in bodily fluid. So not only phosphatidylethanolamine or cephalin is important, but अगर हम बात करें simply ethanolamine की, so it is also considered essential for life because it happens to occur in every cell in the human body as the head, not only as the head group of phosphatidylethanolamine, but it is also present in its free form at various concentration in body fluid. Mammals जो हैं they cannot synthesize ethanolamine, and therefore it is obtained from the diet in its free form or in the form of phosphatidylethanolamine. Mammals जो हैं वो synthesize नहीं कर सकते ethanolamine को. That is the reason it is obtained from the diet in its free form or in the form of phosphatidylethanolamine. That is further degraded by an enzyme that is known as phosphodiesterase to yield glycerol and ethanolamine. Other sources of ethanolamine or phosphoethanolamine in the human body are the degradation of sphingosine phosphate, and this takes place by an enzyme sphingosine phosphate ligase. So other sources जो है ethanolamine और phosphoethanolamine के in the human body, they are the degradation of sphingosine phosphate by sphingosine phosphate lyase, that is the enzyme that causes the degradation of sphingosine phosphate. Plants possesses the serine decarboxylase enzyme that converts serine to ethanolamine, whereas humans cannot do this. So we can say that in humans you have to obtain it through the diet, whereas plants जो है they can synthesize it. How? That they possesses an enzyme that is known as serine decarboxylase. This enzyme converts serine into ethanolamine. So the plants can synthesize this. Plants can also convert ethanolamine to choline. Yeast and human can also catalyze the stepwise methylation of phosphoethanolamine to phosphocholine. But the main and the key difference is that in yeast and mammals, the ethanolamine head group of phosphatidyl ethanolamine is methylated. So as we said in our last line, that yeast and humans जो हैं, they can catalyze stepwise methylation of phosphoethanolamine to phosphocholine. But uh, the difference is that in yeast and mammals, the ethanolamine head group of phosphatidyl ethanolamine is methylated. That will yield phosphocholine. So this will take place in case of phosphatidyl ethanolamine and not in case of free phosphoethanolamine. Two enzymes carry out this reaction in yeast and one enzyme carry out this reaction in human cell. Apart from lecithins and cephalins, that are the two important ones, there are other glycerophosphatides that are found in nature, and their journal formula can be represented as. So this is the journal formula. We have discussed this formula time and again earlier as well. This is the fatty acid attachment site R1 and R2. This is glycerol, this is the phosphate, and there X represents the site and the area where the other groups has to be attached. Like if we are talking about choline, that will attach over here. If we are talking about ethanolamine, that will attach over here. If we are talking about serine, that will attach over here. If we are talking about inositol, that will also attach over here. So whatever phospholipid we are talking about, whatsoever is the functional group, उसकी बेसिस पे जो है वो हम उसको अटैच करते चले जाएंगे. Sorry about the delay. जी. So this is the formula we were talking about earlier and that was interrupted. So coming back to it. ठीक है जी. As we said, the substituents could be serine, N-methyl ethanolamine, threonine, or whatsoever group that has to be attached at the position where we have mentioned X. वहाँ पर जो है ये substituents की attachment होगी. In addition to nitrogenous bases, the substituents may also be non-nitrogenous like phosphatidyl glycerol or inositide. So not only the nitrogenous bases, but other than the nitrogenous bases, they can also be attached at that site, and whatsoever group will be attached over there, we will name them accordingly and they will perform the concerned function. क्लोजली रिलेटेड टू फोस्फोटाइडल ग्लिसरॉल इज कार्डियोलिपिन. कार्डियोलिपिन जो है इसको हम डाइफॉस्फोटाइडल ग्लिसरॉल भी बोलते हैं. दिस इज द बेसिक स्ट्रक्चर ऑफ कार्डियोलिपिन और मोस्ट कॉमनली नोन एज डाइफॉस्फोटाइडल ग्लिसरॉल. ये है जी कॉमन स्ट्रक्चर ऑफ डाइफॉस्फोटाइडल ग्लिसरॉल, जिसको हम कार्डियोलिपिन बोल रहे हैं. कार्डियोलिपिन जो है इट्स अ यूनिक फॉस्फोलिपिड, और वी कैन से दैट इट इज a dimeric structure by comparison with other phospholipid. Cardiolipin जो है it's a unique phospholipid because it has a dimeric structure if we compare it with other phospholipid having 4 acyl groups, 5 chiral centers and potentially carrying up to 2 negative charge. So if we look at the structure जिसको हम कह रहे हैं कि इसको हम कह रहे हैं कि it's an exceptional phospholipid because of the presence of 4 chiral centers, sorry, 5 chiral centers, 4 acyl groups and 2 negative charge. Cardiolipin is present universally in both prokaryotes and eukaryotes, where it represents an important phospholipid component of specific membranes. Cardiolipin जो है वो present है universally in both prokaryotes and eukaryotes, where it represents an important phospholipid component of specific membrane. So it is found almost exclusively in certain membranes of bacteria like plasma membrane and hydrogenosomes, and a few species of archaea, and in mitochondria of eukaryotes. The trivial name cardiolipin. Why we call it cardiolipin? It is called as cardiolipin because it was for the very first time found in animal hearts, where it is especially abundant. So that is a main reason it is called as cardiolipin, and it can be found in mitochondria of all animal tissues and of in the eukaryotic kingdom as well. Um, Cardiolipin accounts for about 10% of the phospholipid of bovine heart muscle and 20% of the phospholipid of the mitochondrial membrane in this organ. So it is substantial component of the phospholipid of skeletal muscle. In most animal tissues, a cardiolipin contain almost exclusively 18 carbon fatty acid, and 80% of this is typically linoleic acid. So बहुत सारे animal tissues में cardiolipin जो है that contains almost exclusively 18 carbon fatty acid, and out of these 18 carbon fatty acid, 80% of that is typically linoleic acid. Among tissues, testis cardiolipin is an exception in that it contains mainly palmitic acid. So as we said that cardiolipin में 80% जो fatty acid है वो linoleic acid है, but जो testis cardiolipin है that is an exception that contains mostly palmitic acid. In the central nervous system, cardiolipin जो है that contains a much wider range of fatty acids that include palmitic, stearic, oleic, arachidonic, and um, decohexanoic acid. So जो कार्डियोलिपिन मौजूद होता है सेंट्रल नर्वस सिस्टम में, दैट हैव मल्टीपल वैरायटी ऑफ फैटी एसिड्स, जिसमें पामेटिक भी आ जाता है, स्टैरिक है, ऑलिक है, रेकिडोनिक है एंड डेकाहेक्सानोइक एसिड्स है. लिम्फोब्लास्ट में जो कार्डियोलिपिन है दैट contains only monoenoic fatty acids. Most animal species have similar composition, but bovine heart cardiolipin differ in that it is enriched in alpha-linolenic acid. As we said, cardiolipin जो है already हम इसको कह रहे हैं कि it's quite different type of phospholipid. So The main difference is, or the main thing is, that it contains fatty acid, various type of fatty acid. जो most animal species है उसमें composition जो है वो similar होती है, लेकिन जो bovine hard cardiolipin है वो differ करता है, because it is enriched in alpha linolenic acid, not linoleic acid. It is rich in alpha linolenic acid. In the cardiolipin of higher plants, linolenic and linoleic acids predominate, and the most abundant molecular species contain 10 or 11 double bonds in total. So अगर हम higher plants की बात करते हैं, तो जो higher plants है उनमें कार्डियोलिपिन जो है उसमें लिनोलिनिक और लिनोलिक एसिड दोनों जो हैं वो प्रीडोमिनेंटली प्रेजेंट होते हैं, और जो मोस्ट अबंडेंट मोलेकुलर स्पीसीज होती हैं, they contain 10 to 12 10 to 11 double bonds in total. So that was all about the different type of glycerophospholipids. The next type of phospholipid में ही जो आता है जिसको हम बोलते हैं जी plasmalogens. Plasmalogens, also known as ether lipid. Why we call it as ether lipid? Because instead of an ester linkage, ether linkage is present in plasmalogen. Plasmalogens are chiefly found in brain and muscles. Um, Plasmalogens are somewhat similar to cephalin or lecithins, but the difference is that the carbon number 1 in plasmalogen is subsequent to the glycerol moiety. Carbon number 1 that is subsequent to the glycerol moiety, that is bonded to it via an alpha beta unsaturated ether linkage in the cis configuration rather to an ester linkage. ठीक है, एक सेकेंड हम जी, दिस इज द स्ट्रक्चर ऑफ plasmalogen. This is the ether linkage. Here is the fatty acid. This is the ester linkage. This is the phosphate group, and this is the area where the subsequent or the group has to be attached. ठीक है जी. So we will continue plasmalogen in the next lecture. Just go through it, and then let me know if you are unable to understand anything, or if you have any questions. We will start our next lecture from plasmalogens.